some of the most important crops that we have today are very recent inventions, for want of a better word. So blueberries, for example, have only been domesticated within the last 100 years. Nowadays, if you buy blueberries, they're this lovely crisp fruit. The wild ones are usually very soft. And that was one of the most important traits, like increased size, but also crisp, because it meant that they could be shipped. So now you can eat blueberries from Peru here in the Netherlands, which would not have been possible with the original wild material, because it would have come and arrived here as some sort of soup.
What impact can a single person have on the world? In this conversation, I speak with plant geneticist Padraic Flood about his mission to domesticate the Aardaker, a small but potentially world-changing plant. The Aardaker has an edible tuber like a potato, high protein content like a soybean, and it can produce its own nitrogen fertilizer directly from the air. If Padraic can increase its yield, this unassuming plant could improve soil quality, feed the population, and help restore vast tracts of land to wilderness. I'm Shane Farnsworth, and this is the Escape Sapiens podcast, supported by the Andrea von Braun Foundation. If you enjoy what I'm doing here, you can have a direct impact on the success of this program by liking, subscribing, and sharing. And now, here's Padraic Flood. I hope you enjoy.
Escape Sapiens.
So I want to start off with a little bit about who you are.
Okay.
So to start with, because you're a geneticist, what is population and quantitative genetics?
Right in the deep end. Population quantitative genetics. So first of all, population genetics is somewhat self-explanatory. It's the genetics of populations. And because most organisms, especially most organisms that we're familiar with, plants and animals, are sexual, so you have genetic flow of genetic information between individuals. The genetic processes, many of them, most important ones, occur at the population level. So natural selection, much of evolution is governed by dynamics at the level of the population. And then quantitative genetics is the study of quantitative traits. So that means you can have a qualitative trait. Classically, one of the Mendelian traits from Mendel would be pink flowers or white flowers. It's either pink or it's white. There is no intermediate. But most traits are not like that. Classic example is human height. There's not just really tall people and really short people. There's actually a continuum of heights, and it's normally distributed. So those are quantitative traits, and they're usually governed by many, many genes. Often thousands of loci are involved. So that's basically quantitative genetics. And most of the really important traits, especially speaking from a plant science perspective, are quantitative. Perhaps the most important one in the world of crops is crop yield. So that's definitely, we would also call it polygenic, but quantitative, yeah.
So you were originally a researcher at an institution, and then later on you worked for, I think it was InFarm, which is a vertical farming organization. But you've left all that now. You're off on your own because you have a dream. Right?
Well, yeah, I have a dream, yes.
So what is the dream, and what's the Aardaker?
Yeah, well, the Aardaker, I suppose, is the dream. The dream, in its largest sense, is to be able to create new crops. And the reason that I think this is exciting and interesting is, if you look at the food that we eat today, approximately, depending on how you count, like 3 species account for the majority of calories consumed globally. That's rice, wheat, and maize. And about 95% of our calories come from about 30 species of plant. That might not be surprising or shocking, but if you look at the number of edible plant species there are, depending on who you ask, and it's not exactly known what the number is, but it's probably somewhere in the order of 20,000 species of plant we can eat. So there's an awful lot of untapped potential out there. Most of what we do at the moment when we think about improving food security or nutritional security is to look at the crops we already have and to try and improve them. And that's a very valuable and valid approach. But there's the other side, which is to look at those other species out there that are edible, to try and bring those into the fold, so to domesticate them. That's what that would be. So, and that, I think, there's an incredible opportunity there, because many of these plants have properties which are of interest to us, whether they're protein rich, like, for example, the Aardaker, and we'll get to that in a moment, or they could be very stress resistant. So they could be salt tolerant or heat tolerant. And we're entering a period of global climate instability. We need more resilient and productive crops. And I think exploring the potential of all those different edible species there is something we should definitely be doing. So that's the larger vision, that's the larger dream, which is to basically be able to do domestication on demand. But you've got to start somewhere, right? You've got 20,000 species of plant. You've got to focus. These kind of projects, like domestication projects, are quite a lot of involved work. So you can't just sort of say, well, let's do all 20,000. You have to pick. And that's an interesting question in and of itself, like which one should you choose? And there is no right answer. There is just, you have to settle on one at some point. And I've decided to settle on the Aardaker. And the Aardaker, we have some here on the table, is a tuberous crop. So in that regards, it's similar to a potato or cassava or sweet potato. So it has underground storage organs called tubers. In this case, root tubers. Potatoes, for example, are stem tubers. And what's special about it compared to other tuber crops is the tubers contain a lot of protein. So cassava is like 1% protein, potatoes maybe 2% protein. The Aardaker can be up to 15% protein of the fresh weight, which is really like a lot more than most of our other crops. And it's able to achieve that because it's nitrogen fixing, which also has additional benefits. So nitrogen fixing means that it forms a symbiotic relationship with rhizobia, a type of bacteria, and through this they're able to fix nitrogen. So nitrogen is abundant in the atmosphere, but it's really not abundant in the soils, and as a result we need to apply a lot of it in the form of synthetic fertilizers or organic fertilizers.
So this plant won't need fertilizers?
It won't need nitrogen fertilizers. And that has a big benefit, both in terms of the energy required to make those fertilizers, if they're synthetic fertilizers, but also these fertilizers often result in pollution. So they get into the water, they get nitrates into freshwater bodies, and that's bad for wildlife. It's bad for people drinking that water. So it's something in Europe, and particularly here in the Netherlands, that we're quite concerned about, which is too much nitrogen. So having crops that can fix that nitrogen themselves means that you get it more sustainably because the plant is providing the energy for this, but also sustainably in terms of you don't get any pollution of nitrogen. And all of our nitrogen fixing crops, we have some of them already. Examples would be peas, beans, soybean. They're all seed crops. And they're great, but there are intrinsic yield limits associated with it being a seed crop. There's a thing called harvest index, which is the percentage of the above ground biomass you can harvest. You can't go above 0.6 because anything higher than that, and there's just so much weight in the seeds that the plant will fall over and you won't be able to harvest it. So there's a physical limit to where you can go. Those limits don't apply to the same way for tuber crops. You can go easily to a harvest index of 0.8. They're underground. They don't need to be held up in the sky. There's a few other reasons why tuber crops are potentially better. They don't need to flower to produce the thing you're harvesting, and flowering is a very sensitive process. If you get bad weather during flowering, you have a big yield hit. You can continuously produce something which is harvestable throughout the growth season, whereby if you're a seed crop, you can only produce the harvestable component after flowering. So if you get bad weather during this period known as seed filling, you also take a big yield hit. So the yield potential and the yield resilience is better for tuber crops. And some of our highest yielding crops are tuber crops or vegetative crops, things like cassava, potato, sugarcane is perhaps the best. But they're all, unfortunately, very low in protein. So they're very rich in starch. So you really, if you want to have plant-based proteins as a large part of our diet, the current tuber crops just don't cut it. Yet Aardaker combines these things. So you have the yield potential of a tuber crop, you have the benefits of nitrogen fixation of a pea or soy, and you have also that yield resilience. Yet you've got lots of protein in there.
Right.
So that's really important.
So what does this do that, for example, soy doesn't do? It's just the yield.
It's the yield potential. I should qualify that because right now it's a wild species, right? We still need to domesticate it. So right now its yields are low. But that is, it's about looking what it can do in the future. So soy is currently our highest yielding protein crop. Based on some data I have now, based on individual plant measurements, this year we have our first field trials. So we'll be able to sort of get a better idea of how accurate this is. But taking rather conservative estimates, already this can produce 40% more protein per hectare than soy. We expect that if we can reach similar yields to potato in 10, 15 years from now with active breeding, we will be able to produce 5 times as much protein per hectare than soy. And that is largely because it's a tuber crop. And all of our other protein crops, as I already mentioned, are seed crops, and they do have those limitations associated with them.
So we get a higher protein yield
Per hectare.
per hectare. It's nitrogen fixing.
Yes.
So at the moment they look quite small. Right? This is not. I can see we've got.
We've got a
what
few.
is it, 4 of them in front of us on the table. They're much smaller than a potato. Are you expecting to be able to grow them to a much larger size, or?
Absolutely, and I have. I mean, I have some that have been up to 450 grams, so like a large potato. This is the wrong time of the year. This is now July. They're harvested in the autumn. These were some I had in storage. Actually, I had them in a pot, so I was able to pull them out. They can get a lot bigger than this already, but as regards to breeding goals, like definitely average tuber size does need to be bigger than what it is at the moment, but also tuber uniformity. So you could have one really large tuber, but you'd probably prefer to have 3 or 4 decent sized tubers.
Okay, so you're actually breeding for uniformity.
Yeah, that is one of the breeding goals. I mean, number one breeding goal is, of course, yield. That is the most important trait. And then protein content is something that we need to keep an eye on. Flavor. Yeah. So flavor depends on what the end use is. So for fresh consumption like this, these have just been boiled just so you can taste them.
Yeah.
But if you wanted to use it as a food ingredient, then the flavor of the tuber itself might not be so important as long as there's no off flavors in, let's say, the protein or the starch, because you break it down and you do extractions. So you could have eventually 2 separate breeding programs, one where flavor is a very important trait and the other one where it's really not that relevant. And then there are other things like shape. So they can be very lumpy and forked. Like you can imagine a forked carrot. That is not desirable if you're selling them as a fresh product where people want to bring them home and maybe peel them or something. It's not such an issue, again, if it's a food ingredient where they're just going into a factory and being mushed up and the protein and starch is being extracted. So, yeah, there are different so-called idiotypes. Idiotype was a term we use for, like, let's say, the ideal plant of a given crop, what it would look like, and that depends on the end use. So if we're talking right now about fresh, like selling it as a potato, effectively, a protein-rich potato, then yeah, I mean, larger tuber size, more uniformity, a nice shape, flavor, protein content is important, and other things like texture of the tuber itself. And then you have all the other things which are really important but not visible to the end user, like disease resistance, stress resistance, overall plant vigor, these kind of things.
It looks exactly, it's like a very, very, very small cassava, right? But it's not so small. So this is the wild plant.
Yeah, this is a wild plant right now. Yeah.
So can we try it?
Yeah, please go ahead. Do you want me to eat it first so you can trust that I'm not trying to kill you here?
I can eat the skin, right? No,
you can. Well, you can, but I think it's nicer without the skin.
It's really nice, actually.
It is. Well, it was consumed as a delicacy for thousands of years across Eurasia.
If I wanted to buy this, I can't buy this, right? It's not a...
Not yet.
Not yet. But it's almost...
Well, unless you... I am selling to a restaurant this autumn, the new Winkel. So if you feel like going to a Michelin star restaurant, they'll have it on the menu.
It's almost, I would say it's like a really, really nice chestnut.
Yeah. So people do say that. It's a bit sweet and nutty, but very mild flavor. And the fact that it's a mild flavor, from my perspective, is a good thing because it makes it easier to use as part of a dish, right? You don't really want a very strong tasting potato because you want to use it with other things, right?
But you didn't add salt to this. Oh, you did?
I sprinkle. I boiled it with some salt.
I was going to say, it's much, yeah. But that's very nice, actually. Okay, so I can see why I can see why potentially you selected this. It's also just something very nice to eat.
I have to admit, I selected it before I'd ever tasted it.
Okay. Because
I started 4 years ago, but at a very small level in my back garden here. And then I had to grow it first before I could get the chance to taste it. So in a way, I had to do it because you can't buy it anywhere. You can't buy the tubers to eat.
Okay, so if this is so good, right, why is it not already domesticated?
Yeah, that's a very good question. I should stop eating this.
Yeah, we're going to be like smacking in the microphone.
When people ask me this question, I always want to take a few steps back and look at the bigger picture, which is, I think you've also got to ask yourself, well, why is it that we have the crops we have in the first place? And I said earlier on about how there are so few species that account for most of our calories compared to the range of species we could potentially eat. And why exactly it is that we eat those species as opposed to others, I don't think anybody has a very certain answer, to be honest. And I think a lot of it is possibly an accident of history, like wherever it was that, like, agriculture started approximately, depending, maybe 8 times independently. So the most famous ones being, let's say, the Fertile Crescent in the Levant. You had in China, you had also in Mesoamerica, in South America, in the Andes, and possibly also in the Amazon basin. And then you also had in Papua New Guinea, and there was almost certainly some independent agriculture occurring in Africa, maybe Ethiopia, and maybe the Sahel. I mean, I think certainly there are crops that originated from these regions. Europe and the agriculture that we have in Europe, which has been very influential in global agriculture because of the way history has gone, is all derived from the agriculture that was discovered in the Middle East, so in the Fertile Levant. So the crops that we have originally in Europe came from there. And so they chose obviously species that were growing in the Levant at the time. And they chose grains initially. And then there's the whole beer before bread hypothesis, which is the idea that initially they were choosing them as a source of alcohol before they even settled. And
do you think that makes sense? But because I just spoke a couple of weeks ago to Edward Slingerland, and so he talks about this quite a lot in his book, and also we did in the podcast as well. So do you think we domesticated grains for beer or for bread, and then beer came later?
I think beer first makes sense to me.
Why?
Well, if you were just going on first principles and your initial goal was, we need food security, and I've got this great idea that instead of going around gathering these plants, we could control their cultivation locally and have our food supply, I think you would go for something like a root or a tuber. Because, you dig that up, you've got a bunch of calories there, it's easy to access, and it's easy to process. You just heat it up, or maybe even eat it raw if it's something like a carrot. With a grain, it's a grass. Like, why would you go and say, oh, we're going to go and collect these grass seeds? I think that just seems like an awful lot of hassle and work. And then you've got to process them into bread. Again, a lot of hassle and work. And it just doesn't seem like, if your original goal was, let's reorganize our way of getting food and start cultivating it, that would not be your first port of call. Whereby if instead you're like, hey, if we get these seeds and we let them ferment a bit, we can all get drunk, and that's loads of fun. And you're still focused on being a hunter-gatherer, but over time you're like, yeah, for our annual shindig, we want to make sure we have a supply of these grains so that everybody has enough beer. And then eventually, maybe over time, who knows, I speculate, but maybe some shaman or something took responsibility for making sure that there was enough of these grains, and then cultivation started to evolve out of this to ensure that there was a supply. And then eventually, perhaps they said, oh, well, you can also eat this. And then bread came about. And eventually then, well, as they say, the rest is history, but we had fully fledged agriculture at some point. And also, if you look at these ancient societies, you also see beer being a really major component in the Sumerians, in the Egyptians. And then I think another, for me, really good case is maize. So the wild ancestor of maize is teosinte. That is not at all looking like modern maize. And it doesn't look like a very attractive food. It's got these really hard seeds. You need to take a hammer to crack them open. It's not an obvious choice if you want to make, like, I don't know, whatever, some kind of porridge or bread-like thing that people make from maize. But it's perfectly fine if you want to make beer, because it's easy to sprout the seeds. So I think that hints that maybe the original use for some of these crops was not food. So coming back to your original question, well, why not the Aardaker? Well, the decision-making process behind the choice of our most important crops may not have been guided by anything like food security, the way we think today, but rather booze security, for want of a better term. So I think, first of all, and then, there's a degree of contingency with how things progress. Like historically, there's always the what if question, but once they were being used and being cultivated on scale, and you had wheat and barley and maize and rice in different parts of the world, and you learn from your parents that this is what you're going to grow, there's not an awful lot of... it's not really something that people go out and say, well, you know what, let me go out and just try something different than everybody else has done. Now, that has happened, but it's usually happened for things like vegetables, where people were like, we want a new flavor, we want to add some... But the core commodity, the main calorie source, remained very stable throughout all of this time. And the only major innovations, if you will, in the main calorie source in people's diets was when there was contact with another independent source of agriculture, like classically the potatoes. So there was contact between the Middle Eastern package of agriculture and the South American Andean package of agriculture. And it was, well, these potatoes grow great in Europe. And actually, they had some of the attributes that I've already spoken about with regards to a tuber. They brought a whole load of additional food security to Europe. And that actually that changed history, because European population grew in a way that was never possible before. And then, unfortunately, there were famines associated with this, which then led to mass emigration to America. And this is getting a bit off topic. But I think the reason that we have the crops we have is more of an accident of history. And out of the 20,000 species we could choose, the fact that we have the ones we have is, well, it is the way it is. But there is no reason to think that the remaining 19,500 or whatever it is don't have something really useful there for us. So I think the question is less, why haven't we domesticated, or if the Aardaker is so great, why haven't we domesticated? It's more, why have we domesticated so few in the first place? I think it's, as I said, an accident of history, and there's just so much potential there that we, especially now with modern technologies, can accelerate this process. Because normally domestication is a very long process, but I think things have changed.
I'm surprised that you selected the Aardaker before ever tasting it. Right, so
well, I read that it was very tasty.
Right.
And I had to believe them. And that was actually, the reason I believed that was there's not a lot of literature on it. And most of the literature is ethnobotanical.
And you actually found it, right? So the plant
it grows native here in the Netherlands. It ranges from the Netherlands to Lake Baikal, and from Spain or Italy up to Sweden. It's particularly common in Eurasian grasslands.
But it was endangered at some point, right? I did a little bit of reading.
Well, so it was never endangered as a species as a whole. It was locally considered endangered in the Netherlands. I don't know whether that was also because of a lack of data. But in 2017, they took it off the endangered plant list. It's not rare around here, and it's certainly not rare as a whole. I mean, if you go to Hungary or Ukraine, Russia, it's really common. It's not a rare species at all in these parts of the world.
One aspect of the story that you pointed out is that, so this is a pea plant, essentially.
Yeah, it's in the pea family, the legume family, yeah.
So why is it that you're harvesting for the tuber that's underground as opposed to the pea, right? So if I was thinking of, I'm going to be breeding this or cultivating this pea plant, I'd be thinking about the peas first up.
Well, that comes back to what I was saying earlier about the intrinsic yield potential of the tuber. And in this case, these particular seeds are rock hard. And in this particular genus, Lathyrus, there's another crop known as the grass pea, Lathyrus sativus. And this is one of those ancient crops that's been around for at least 8,000 years. And it's a pretty incredible species. But unfortunately, inside the seeds is a chemical called ODAP, a non-essential amino acid. And if you have a poor diet, so if you're food insecure, particularly if your diet doesn't have a lot of methionine in it, another essential amino acid, and you eat a lot of this ODAP, you can develop a disease known as neurolathyrism, which is irreversible and very unpleasant. Historically, grass pea was cultivated throughout southern Europe. It's still cultivated to some extent in India and Ethiopia. And when all the other crops die, often this is the only one left. And then you have this perfect storm situation, because the reason the other crops died maybe was excess heat or drought. And when you've excess heat and excess drought, the plant accumulates more ODAP, and the people have less other food. So they end up eating only this, or almost only this, and it's got extra ODAP than it normally would have. And then you get a perfect storm where people start to develop this disease.
And what does it do, this disease?
The neurolaterism, it's... I forget actually the details of the symptoms, but I do know that it's very unpleasant. But it degrades your nervous system in some way, I think.
So your brain as well.
Yeah, I think you lose mobility in your limbs, and then you eventually die. Yeah, it's not something that you want to happen to you. However, people with a rich diet, it's not a risk, really. And there are efforts actually to bring this crop back into the fold. So there's work being done in the John Innes Institute in the UK to create zero ODAP Lathyrus sativus, grass pea, and also work being done in India on this, because the crop has such an incredible resilience and potential for food security. And it's also very nutritious, except for this one amino acids. It's very rich in protein, the seed. It's a very rich food. It's just that you shouldn't eat only it and nothing else. But if we can create zero ODAP, then that risk would be gone. Now, with regards to the Aardaker, there's zero evidence that there's ODAP in the tubers, although we are double checking this, of course. But there's possibly ODAP in the seeds. So that makes them also not so attractive. But it's a perennial, so it doesn't produce its seeds very uniformly. It doesn't lend itself very easily to being a great seed crop, to be honest. What I can say is that the leaves themselves are also edible for humans and livestock, and they have a similar feed value to alfalfa or lucerne. So that is an additional revenue stream that the crop could possibly give, which is before you harvest the tubers, you would cut the tops and make that into hay or silage or whatever, and feed it to animals. You could feed it to humans, but it's probably at the end of the season going to be a bit rough and people won't really want to eat it. But
so you do that before the seeds form?
So it wouldn't really matter whether the seeds are on there or not, but the way that we're currently growing it, we would be planting seedlings or seeds and then harvesting tubers that year, and they don't flower a lot the first year anyway, so you wouldn't expect a lot of seeds.
And why should the plant have the poisons in the seed but not in the body of the plant and the tuber?
Well, seeds are really important for a plant's survival. So plants often accumulate toxins in their seeds to protect them from beetles, they don't want their seeds to be eaten. I mean, in general, they don't want to be eaten, but toxins cost energy. So they tend to be pragmatic about where they invest these things. And the tuber is hidden. So that's its survival strategy, is to hide away underground. And they keep them quite spread out. So that's also a trait that, from a breeding perspective, we need to work on, which is to encourage the plant to put all its tubers close together. So if you look at other tuber crops, wild ancestors of potatoes, the potatoes are on these very long stolons and super spread out apart from one another, but the domestic forms of the potatoes are clustered together, which makes harvesting much easier. So that is an important trait for us. So so those the plant solves it's the problem of how do I avoid getting eaten in different ways in different tissues. And with seeds, often, not always, they have a lot of toxins in them. And that is true for not just in the Lathyrus genus, but in many, many different plants. Seeds are toxic.
So before we talk about breeding, what could the impact of this plant actually be? Because the way you talk about it, it doesn't sound as though money is the first thing that you're seeking when you're developing this crop, right? It sounds like you have, for sustainability or for some other reason that you're developing this. So what is that? Yeah,
so for me personally, the primary driver here is the impact potential. And that's because originally I come from academia. And, back in 2020, I was supposed to be taking time to write a grant, and I had a gap between two contracts, which was March and April 2020, and I was going to write this grant, and I was very dedicated to the academic track, and I was studying how plants adapt to extreme environments. I was doing a lot of this population and quantitative genetics we spoke about earlier, And at the time, my children were a 1-year-old and a 2-year-old. And then, well, the pandemic happened and there was no daycare, and you can't write grants when you're in the presence of a 1-year-old or a 2-year-old. So I had to put that on hold. And I thought naively, this is all going to be over rather soon, and focus on being a parent. And during that time, I came to realize that I think I was very naive. And I was naive in the sense that I thought, well, this pandemic is a disease. We've dealt with diseases before. We've got a lot of experts on these things. It's a common foe globally. It doesn't matter whether it's China or the US or Europe. We're all up against this disease. So we're just going to get together. There's going to be adults in the room. They're going to listen to the experts, and we're going to have a coherent and unified policy. And that's not what happened at all. I mean, people start politicizing whether you should wear masks. There was all the anti-vaccine stuff. And I thought, my God, this is such a mess. And there's so little agreement even amongst the leaders of the world about how to deal with this. And I thought, relatively speaking, this is a simple problem compared to, for example, global climate change and the sort of damages being done to our ecosystem as a whole. And I'm thinking, looking at my young children, and I'm thinking, oh, I think I need to do something with my time, which is contributing towards a more sustainable future. So I decided to shift my career away from studying how plants adapt to high altitude ecosystems in the tropics, which was lots of fun and very interesting, but not so applied, and think about how I could use my knowledge and skills for something more applied. And I've always loved genetics. That's what I've done. And breeding is basically applied genetics. So then I thought, well, how can I use breeding to try and create a more sustainable food system? And I had always had an interest in wild food as a hobby and in plants in general. So I knew about many of these species as wild food that grew around. And I always thought that there was a lot of potential in them. So I decided I would do that. So I thought, right, I think there's really something here that could improve the sustainability of protein production, because protein is possibly the most unsustainable macronutrient, the others being carbohydrate and starch. But because of the need for nitrogen and because of the polluting aspects of nitrogen, which we discussed already, protein is the one that there's a real bottleneck on. And then there's here regionally, there's this desire for a protein transition away from animal to plant-based proteins. And I remember thinking, there is no good alternative to peas and nuts in terms of a plant protein. So yes, you can have these processed derivatives of them, let's say the plant-based burgers and things like that, but there's nothing like a tuber, which is basically a potato rich in protein, which has that culinary versatility, right? So you've tasted this, but you can also, that was just boiled just so you could taste it in its plainest form. You could easily mash that, bake it, put it in a stew. You could do any of those things and make fries out of it. I've done all those things, make crisps, and you can do what you want at home in your kitchen. So I thought that initially was what I thought was really great. But then I came to the understanding that getting people to change their purchasing patterns and buy a different kind of potato is a very slow process, and it's unlikely to happen very quickly. So then I started thinking more about its potential as a food ingredient. And then that comes back to what I spoken about earlier about the yield potential of tubers and the ability to produce more protein per hectare than any of our existing nitrogen-fixing crops. So that then became very much so my focus in terms of aiming towards that impact. But I decided to move away from academia to the private sector because I actually thought if you can make a business out of this, it's much more likely to scale rapidly to something that will actually make a difference to the sustainability of food systems. If your product is actually the tuber or the food ingredient instead of a paper or a publication, and that's your aim and your goal, well, then you're more clearly aligned with the impact. So I do have to think carefully about how I make this into a profitable business, because I need investment, and that's at least to get something like this kicked off. And so far I've succeeded there, so hopefully we will continue. So it is important that it's profitable, but because of first of all, it tastes good. It's good for a premium product at the moment, but in the long run, it has incredible potential, I think, as a really sustainable source of food ingredients, particularly protein.
As a staple?
Yeah, a commodity, yes.
So when you say protein, that can mean a lot of things, right? So how does this compare to, say, a steak or to the protein that you'd find in a soybean or in milk or something?
So the honest answer is I don't know yet, because we are actually right now checking the amino acid profile. So that would be one of the first things you want to know, which is how complete is the protein. So does it have all the essential amino acids, and does it have them all in the right ratio? So meat, for example, is a really high quality protein from that perspective. But the problem with meat is the sustainability of the protein production itself. And you can combine plant proteins to make something which is more complete. And that's what people have done for a very long time, classically combining pulses with grains because they complement one another's amino acid profile. So right now, I don't know for sure what this is going to be in terms of amino acid profile. From a sustainability perspective, it compares well, or it has the potential to compare very well, of course, with meat, although almost all plant proteins perform well compared to meat. But also, I think it has the potential to perform better than many of our existing plant protein sources.
So then when it comes to breeding, so if we talk about breeding now, what are you doing? So you found a wild variant on the side of the road, you've picked up some seeds, or did you originally dig up the tubers? How did you...
Well, I the very first time I came across one was in spring, so I took a cutting because I was too excited to wait until the seeds came, and I grew that.
In water? Or
I just rooted it, yeah. But yeah, normally I collect seeds because you're not supposed to dig up plants publicly. And then here, you need to be careful with permits and things like that. So also I've sourced some of my germplasm from, there are these germplasm collections in the USDA and in Germany. So, because you need to make sure that everything you get in the proper manner, Nagoya compliant and all of these things. So I sourced the germplasm, and then, I forget where you're going with this.
So where I'm going with this is, so then you want to breed this, right? Yeah. So what does that look like? So you start off with a plant which is completely wild. How do you go from that to something that you can actually sell?
Yeah. Well, so luckily in this case, you can sell it already, as I would hope you would agree. Like you'd be willing to eat that.
Absolutely.
So that means that right now I kind of, the surplus of my breeding program I can kind of sell, which helps me at least cover some of my costs. But obviously what you want to know is how do you get it so that it's high yielding and a farmer actually wants to grow it, and I can then sell the seeds and sell
and so genetics. That you can use potentially the same equipment that you'd use for a potato.
Yeah, exactly. So you need to breed it so that it's agronomically feasible and economically feasible to be grown on a larger scale. And there are many ways to do plant breeding. There are many different kinds of protocols, which depends a lot on the biology of the plant that you're dealing with. In this case, I'm doing a thing called recurrent mass selection at this moment. I will be, now that I have some funding, I'm going to do something a little bit more elaborate. But recurrent mass selection is one of the most basic forms of breeding. And it's probably, in a way, how domestication started originally. And it's basically you grow the plants. They're all different from one another, just like me and you are different from one another. They're all genetically unique. And you pick the best ones, and best being usually things like higher yielding, larger tuber size, better tuber shape. You can create a thing called a selection index where you can put all those traits you're interested in and then also rank them or give them weights based on their importance. And then you can go out and measure all those plants you grow and say, well, these are based on my selection index, these are the best performing ones. So I'm going to bring those through to the next generation. And since some of that variation that you're looking at in the field is genetic, it's not just environmental, then the following generation will also perform a bit better than what you started with. And then you just do this iteratively, and over time, the population as a whole will improve for the traits that you selected for yield and shape and things like that. Now, that can be a slow process. If that's all you do, you can speed it up somewhat by just increasing the sample size. So it's a numbers game. The more, if you've 10 plants and you take the best 2, chances are they're not going to be amazing. But if you've 100,000 plants and you take the best 2,000, those will probably be really good, those 2,000. So, and then you can improve trait values in this way. And then there's a few things you want to pay attention to, like you want to avoid loss of genetic variation, because that's the fuel of your breeding program, the fuel of evolution actually as well. So that's really important. And then nowadays you can incorporate new tools. So recently, genome sequencing. So is that I don't know if I should explain that. So basically, the ability to decode a genome, its basic sequence, has become very affordable. There's been great advances in new technologies so that you can take these sequencing technologies and apply them to any organism you wish. Before, that was very difficult because it was very difficult to do a genome assembly. The original genome assemblies 20 years ago took teams of scientists working for years and years and years and hundreds of millions to make. And now you can do one kind of almost in a weekend for a couple $1,000s. So it's just, there's been an incredible advancement in those technologies.
So can I stop you there? So you're going to have, say, 1,000 that you hand select for size or something along those lines, and then you'll do a genetic testing of 1,000, and each one costs 1,000. This sounds
no. Yeah, that's a good question. First, what you do is you need a genome, which is basically serves as a reference point. So that's the thing that's expensive. So you create a de novo genome assembly, right? So now we have it. We've published it actually, but there's an Aardaker genome published, and we're updating that reference genome. So that's actually, this plant we're eating here is possibly going to be the one that we choose as the reference genome. So that individual, and we'll sort of throw the kitchen sink at it in terms of genome sequencing. We'll use all the best technologies. We'll have a really high level genome assembly of this, all the way to chromosome level, phased, everything, meaning we know what chromosome came from the mother, what chromosome came from the father. And then this becomes a reference to which we can use much cheaper technologies, which we would call genotyping, where you just maybe have a few markers scattered across the genome, so a few thousand might be enough for you. And then you can use that reference genome to position those markers, and then you can genotype thousands of plants for a much more affordable. So instead of it costing 1,000 or 2,000 euro to do the genome assembly, or maybe even a little bit more, it's more like 5 to 10 euro to genotype a sample.
I see.
So then you can do many more. So for example, you said, right, okay, this year now in the production field I have about 20,000 genotypes. I'm going to take the top few percent of those, and a number of those I will genotype. And I might even choose to do more expensive genotyping, so we call that resequencing on some of them, if that makes sense. And over time, because I keep measuring them and I have their phenotype data, and I keep genotyping and I have their genotype data, I can start to link these two data sets. So we call that a genotype-phenotype map. And it's basically a multiple regression problem, so machine learning or AI, if you want to call it. So these statistical genetics is advancing a lot. And that means that at some point when my data set is large enough, just based on the genotype alone, I can start to predict what the outcome will be in terms of phenotype being in this case like yield and protein content. And that's really valuable for a number of reasons. It means that, let's say I want to speed things up, I can do a thing called speed breeding, which is I grow the plants under artificial environments. So let's say in a greenhouse or a growth chamber. I expose them to conditions which forces them to flower after a very short period of time. So that means that I can get, instead of at the moment, one generation every 2 years, I can go from seed to seed in, let's say, 12 weeks, right? That's a great advantage.
Just by changing the environment?
Yeah.
Okay.
So you can basically make the plant think that it's experienced spring and summer all within a very short period of time. So there's plants are sensitive to these environmental cues.
Yeah.
And you can also, you exaggerate, right? You do things like a 22-hour day, which the plant would never experience in nature. And then it kind of gets, let's say, overexcited and thinks, oh my God, I need to flower. And yeah, each species requires a slightly different protocol. Some species require short days, others long days, but you can work that out, right? And this has been well described. And I've already got a relatively decent protocol for Aardaker.
Wait, so how many years can you get in a year?
Good question. Best case scenario, I reckon I can get 5 generations in a year, which will be the equivalent of 10 years normally out in the field.
Okay, so if I understand what you're saying, so you start off with just out in the field, and you'll pick for size, you'll do measurements of the amount of protein somehow in the tuber, and then I guess you taste them as well.
Hopefully.
Taste them,
and then... You cannot necessarily do that on everything. Because it's really there's only so many tubers you can taste and discern at some point, and it's expensive to pay people to do those things. But yeah. But
the end of the story is that you can then map those characteristics, the phenotype, to some signals in the sequence, the DNA sequence that you have.
Genetic markers.
Genetic markers. And then you, inside the lab, can send the plant through several generations in one generation, let's say, or in 1 year.
Yeah, in 1 year, several generations in 1 year.
But you're then looking specifically for those markers.
Yeah, because of the fact that you force the plants to go through its life cycle very quickly, the phenotype that it expresses is kind of uninformative to you, because it's not at all going to be the kind of phenotype that plant would produce in the field. So if you don't have the ability to predict phenotype from the genotype and you do speed breeding, you could inadvertently breed for something which is very poor performance. But if you can accurately predict the outcome, phenotypic outcome under production conditions, field conditions, and you can force the plant to go through many generations in the lab, let's say, or in the growth chamber, that means that you can increase the amount of genetic gain, which is what we call, so how much the yield would improve per unit time, massively. But if, as I said, if you don't have the ability to predict that from a sequence, probably you'll end up just breeding for something that flowers very fast, which isn't necessarily useful, right? Because it might not produce tubers anymore,
right?
Which is, so without the ability to use that, having those molecular markers, those genetic markers, the speed breeding protocols don't really work. So these things help one another, right? It's a confluence of technologies. We've gotten much better at creating these artificial environments. We've been able to make them much cheaper. LEDs are cheaper. Climate control is cheaper. Like the vertical farming industry as a whole has helped bring those prices down. That makes it easier to do speed breeding. The genotyping is cheaper than it's ever been before, which makes it easier to be able to build this genotype-phenotype map and be able to make genomic prediction. So you can now do many generations in a time span which would just never have been possible before. And you can do it on a much larger scale, because if you think about it, if I had a growth chamber, and again, growth chambers, they're cheaper than ever before, but they're still not cheap, right? So if I want to grow a large population of plants all the way up to maturity, I can probably only grow a few hundred genotypes in a growth chamber at best, but I can grow maybe 10,000 seedlings. Now, if I can just do a leaf punch on each one of those seedlings, send it off for genotyping, get that information back within 2 weeks,
then you can cull the others.
I can just get rid of all the others. So that means you also can play the numbers game at the same time.
Because I was going to ask, why do we need a geneticist to do this? Because if you're really just out in the field, just selecting, say, the biggest tubers, and that's all you're doing, then we don't really need you for this program.
Yeah, which historically has how it's worked. I mean, the first farmers have made incredible progress, right? I mentioned maize before because it's an excellent example, but if you look at modern maize and the wild ancestor, they look nothing alike, and that is purely selection by, like you've described, people looking out and saying, hey, this is an interesting one, let's keep that. And that works, but it takes a long time. And right now, with the way our world is, we don't really have time. At least I think we need to be working, we should have started working on this at scale long ago, and we haven't, because we're not always great at organizing ourselves, apparently. So we need to make this happen as quick as possible. And then bringing in, and it's not just a geneticist, but you also need basically a data scientist. That's what I've described, is a lot of this is, you might call them a genomicist or a bioinformatician or something like that, but those are the people with the skill sets to be able to process these large data sets, because the data sets get quite large. I mean, the genome of Aardaker is 6 billion base pairs long, so it's twice the size of the human genome. If you have several thousand individuals' genomes, well, you can imagine the data sizes. And then if you bring in highly multidimensional phenotype data, so everything from yield to protein content to flavor and metabolites and whatever else it is, disease resistance, and then you're trying to do an analysis, a multi-trait analysis and all of this, you can see how very quickly this gets rather heavy, and then knowing how to process large data sets efficiently becomes important.
How do you know, this is completely out of field for me. So one thing that I don't understand is you might have a phenotype which has a large tuber, let's say, but that might have many changes in the genome compared with something that has a small tuber. How do you know, if you're only sampling, say, 1,000 plants, how do you know that it's that part of the genome that's really important?
Yeah, so first of all, you might not figure that out with only 1,000 plants. So you may need more, depending on the design of your population and things like that. But it's also almost never a single locus. So earlier I mentioned about qualitative and quantitative traits and quantitative genetics. So usually there are thousands of loci involved. And then
loci.
Positions within the genome.
I see.
Yeah, so markers or whatever. And I specify loci as opposed to genes because it's not only genes that impact phenotypes. You can also have like basically regulatory sequences which are not inside the gene but promoters and stuff that can have a big phenotypic impact. So that's why we talk about loci specifically. But so it becomes a question of signal to noise and statistical power. So the smaller your sample size is, the less accurate and also the fewer of these loci you will be able to find. A great example is human height. When human height is highly heritable, meaning that tall parents tend to have tall children. I think this is something we all recognize and can see. There is an environmental component. So within the last few generations, certainly in Europe, we've all gotten taller because our diets have got better. That's not genetics, that's environment. But within a cohort of individuals, the heritability of height is very high. So that usually means that your signal to noise is very good. And it's also a trait where measurement error is small, right? It's very easy to measure somebody's height. And we have huge data sets. And initially, when they started trying to untangle the genetics of human height, using a similar approach, this genotype-phenotype mapping I've just spoken about, they used an approach called GWAS, genome-wide association mapping. So they knew that they'd mapped some of the loci responsible, but only a small fraction. And a lot of the heritability, the genetics, was missing. And it became this great, I would call it a pseudo-controversy, which is where's all the missing heritability? And in the end, it turned out, well, you just need larger sample sizes. And when they got up to 500,000 people, they'd accounted for most of it. And now they're up to like 5 million people or more, where they've got the height and the genome sequence data. And basically, they can map all the loci. And there are thousands and thousands of loci that can affect height. But the loci themselves have different effect sizes. So some of them, whether you've got one allele or another allele, alleles being gene variants, can have a big impact, and it can change your height by a couple of centimeters.
And you're more likely to see those, I suppose.
Those are much easier to pick up, of course. There is a confounding factor, which is if they're very rare in the population, they can have a big phenotypic impact, but you might not pick them up because of the fact that they're not a very high frequency allele. So the ones that are easiest to pick up are the ones with a relatively large effect that are at a pretty high allele frequency. So there's the A and the B allele are both at a pretty high frequency. But you can get all sorts of other combinations which require larger sample sizes in order to pick up. So obviously the ones with very small effect, where they might only affect height by a millimeter or something. And then you have situations where they also interact with one another. So that's called epistasis. So genes can interact. So if you've one allele or A at a particular locus and B at another locus, that could make you even taller than either one alone, for example, or even shorter than either one alone, depending. So there's getting around all of these complications. One of the ways that we found that most successful is basically brute force, so larger sample sizes. Now with plant breeding, there's a bit of an advantage. You have more control, so you cannot force people to all be in the same environment, right? There's that environmental noise with height and make everybody eat the same food or whatever you would need to do. But you can with plants, you can exert a lot more environmental control. And then because you can effectively breed from generation to generation, if you have a false positive, like you think something's important for improving yield, but it isn't, and then you run the selection for a few generations, and each time you recalibrate your model, every once in a while you'll grow something up properly and phenotype it and genotype it, those false positives will go away. They will just get weeded out, so to speak. Whereby what you're much more worried about is the false negatives. So your type 1, type 2 error preference in plant breeding is very different than that in human genetics and medical genetics, where we really don't want false positives because that could lead us down the wrong route for developing medicines and things like that. So we're very worried about them. But that's really not a concern for me, for example. I'm worried about false negatives. So yeah, there's a few different considerations, but ultimately, finding those loci and mapping them, it is largely a numbers game. There are important things about the accuracy of your measurements, ensuring good signal to noise, and there are things you can do to control that, growing them under a controlled environment, but that gets expensive. So you may instead choose to do what I'm doing, which is try to keep it cheap, grow it in a field, which is cheap, suffer the fact that, or the consequence of more noise, so reduced signal, but then try and brute force it. So increase your sample size and say, well, we'll just keep doing this for a few years and we know that we'll pick up signal. And over time, that signal will give us results.
And so how many years are you going to run in the field before you do this speed reading protocol where you can...
I will run them in parallel.
I see.
So I've done the field now because up until very recently I didn't have funding, but now that I have funding, one of my high priority things is to set up the speed reading protocol.
But don't you need to do the field first so that you know what to target for the speed reading?
Good question. Good point. Yes and no. So developing the speed breeding protocol in and of itself, I mentioned I've already made some progress, does take time. So getting just what is exactly the right environmental recipe to make these plants flower as soon as possible. Can I take immature seeds and grow them? And if so, can I, because you could, like the seed development itself can take 5 weeks. So if I can make the plant flower in 5 weeks, but it takes 5 weeks for the seeds to develop, that's giving me a total of 10 weeks. But if I could 3 weeks after the seeds develop, pop that seed out, it would normally die. But if I stick it on some agar or something, I can shave off 2 weeks off my generation time. And of course that accumulates. So just refining those protocols takes time, which means you should start immediately. So that's why I would start already. And then there are other things like trying to create inbreds, which is very important. And that you can kind of do with speed breeding already without the need for the sequence data or anything like that. The sequence data would help. But the question, like, would you not wait until you have your genomic prediction model in place? Is, well, yes, you will be able to use the speed breeding more efficiently and to its full capacity when you have a good genomic prediction model. But I wouldn't wait for that to start getting my speed breeding protocol going. And yeah, so now we're sequencing 67 genomes, whole genome sequencing, which is quite expensive, but that's an investment and that will be a baseline data set. And we're measuring those plants for a lot of things. The elite germplasm that comes off the field this year, the best performers, will also get genotyped, probably not whole genome sequencing. And then I'd say by next year, we will start to have not a great genetic prediction model. It'll be kind of shoddy, but enough to start with the speed breeding and be sure that we're probably going somewhat in the right direction. And then you constantly recalibrating the model, right? Every year you're putting out germplasm under production conditions, growing and genotyping it, and then feeding that back in. And I'd say by 2028, we're going to have a relatively decent genomic prediction model and a speed breeding program so that we can really crank the generations along very, very fast.
And so this is also why you need to do it in parallel, because you need the plants grown in the field so that you can get the map between the genotype and the phenotype. So that's absolutely necessary to keep that in parallel.
Yeah, and also, well, the field allows me to, I would keep the field anyway, even if I had loads of money and just do everything in a grow chamber. The field also allows me to refine things which have nothing to do with the breeding program, but are very important for the crop development, which is the agronomics. So how do we grow this? How do we harvest it? Let's make sure that
also
it fits
resilience, I guess.
Yes, but just the prosaic, like, how do you grow the crop? If it's not performing well, if it's too wet or whatever, what are the interventions you can make? Because ultimately you're going to want to sell to farmers, and they don't want to have to figure this out all for themselves. They want you to come to them and say, this is how you grow this thing so that you get good results. And right now, we don't know the best way to grow it, right? We're trying different things. This year I had 3 different fields. 2 of them failed because I tried something and it didn't work. So one of them's working, thankfully. Otherwise I'd really be in trouble. And these are the kind of things that you just need to keep doing. And that you're sort of limited in that your learning curve is 1 season a year. And that's it. Unless you're willing to also try and do a season in the Southern Hemisphere, which if budget allows, I'm fine to go to South Africa or Chile and then have 2 seasons in a year. But that's about all you can do under production conditions.
You mentioned before the conversation that ultimately you want to sell to the farmer. And at the moment what you're doing is you're producing seed and you're replanting the seed. And ultimately you want to also do that with the sales, right? You're not going to sell tubers like you would with potatoes, but you'll sell the seed and you grow from that. Why? So my picture is that there's going to be more variance in what the farmer gets if you're selling them seed. So why sell seed as opposed to selling the tuber?
Yeah. So, good question. So right now, I sell the tubers not to the farmers, but to restaurants, just because we have to get the business off the ground, and the crop doesn't exist, right? So if nobody's going to grow it if there's no market. It's a bit of a chicken and egg problem. There's no market because nobody grows it, and then if there's no market, nobody wants to grow it. So you kind of have to push this yourself. So right now, that's why I actually grow and sell tubers. But the long run, it's the goal to sell the genetics, not the food, right? And then your question is, why not sell the farmers the tubers, get them to plant the tubers, and then you can very easily have a very uniform crop because you can clonally propagate the tubers. And that's exactly what they do with potato, right? They don't plant true seeds, they plant actual tubers. But the potato industry as a whole, this is a bit of a headache for it, because when you clonally propagate plants, you have a lot of challenges associated with that. On one hand, you have a higher risk of disease, because viruses are passed much easier via vegetative tissue than they are via seeds. So that means that your seed potatoes, these little tubers that you plant, have to be grown under very clean and careful conditions, either in a controlled environment, or often they grow them on islands and places where there's low disease pressure. Yeah. So then they have to ship those tubers to where they're actually going to be grown. So this costs a lot of money, and you're ultimately shipping something which is 80% water. And it needs to be kept cold so that it doesn't go bad. It means that certain parts of the world where the supply chain is not so well developed, famous places like sub-Saharan Africa, it's really hard for them to get good quality planting tubers. If the farmer decides to save his potatoes and replant them the next year, he will probably get a lot of problems with disease, so they don't want to do that. So there's a lot of push and a lot of interest in having seed propagated potatoes, and there are companies like Solinta and more recently Ohalo Genetics that have said that they are going to solve this problem, because it's widely recognized that this would alleviate a lot of these issues and bring a lot of value ultimately to the supply chain. So if you look at the problems that the potato industry has faced with regards to disease and supply chain, and you're starting out from scratch like I am, the obvious thing is to say, well, okay, we can propagate it via tubers, and we might do that as a stopgap at a small scale initially if we have a really good clone which is just performing amazingly and we want to get our production up. But it would not be smart to make that your long-term goal. Instead, you want to be able to sell the seed to the farmer because of the reduced disease issues, and because a pack of seeds, like a bag, a few kilos, will sow and you can plant several hectares, whereby you'd need a truckload of tubers to plant the same area, which would be a real difficulty. And then you asked about uniformity. So obviously, if you're planting tubers, you can plant a clone, so there's complete uniformity from a genetic standpoint. With seeds, you can achieve that as well through either generating what are known as inbreds. So these would be completely homozygous plants, although they are not always the best because they often have inbreeding depression.
They're less resilient.
Yeah, they're just weaker. They're a bit sick genetically. This is not the case for all plants. Some plants are perfectly happy to be inbred, like wheat and rice and tomatoes. You can inbreed them and they're fine.
Do you have the problem here with the
Well,
Aardaker,
it's not entirely. There is some inbreeding depression in Aardaker because it's naturally quite outcrossing. But on the and that's something, for example, maize has a lot of inbreeding depression. Potato also. So making inbreds in these species has been challenging, although they have succeeded in the case of maize.
But wait, so just a quick question. With wheat and maize, that doesn't have a tuber, right?
No, but whether you have inbreeding depression or not has nothing to do with whether you're a tuber or a seed crop.
But my question is rather, how do they do the propagation then? They need to use the seeds.
Yes, they've always used the seeds. But in the case of wheat, when you buy wheat seeds as a farmer, you usually buy an inbred. So it's all one clone. And if you save the seeds, you will get the same crop the next year. Right? Which is kind of good for the farmer, not great for the breeder, because the breeder then needs to chase up people if they spend years developing these varieties, could be a decade or more, and then they have to enforce their
intellectual property.
Yeah, intellectual property rights, which in some parts of the world works well. Depends on many things. So breeders are always on the lookout for, can they create an F1 hybrid?
Sorry, what does F1 mean?
Filial one, so the first generation, filial being son. So you will have two parents, parent one and parent two, and the offspring is the F1. So you're the F1 of your parents, right? In plant breeding, what we try to do is we try to create homozygous parents, inbred parents. So they might be a bit sick in the case of an outbreeding species like maize.
So the two parents are highly related to each other?
No, not at all. They're completely unrelated to each other, but they themselves are completely inbred.
I see.
And actually, that's key that they're unrelated to each other. Because then when you combine them, the offspring is highly heterozygous. So it doesn't have any of the inbreeding depression. Super vigorous. You get a thing called hybrid vigor or heterosis. But because the parents are homozygous, it's heterozygous but uniform. And uniformity is really important in a crop cultivation context.
Wait, so you just sort of reading forward in the conversation. So you're going to be doing the breeding twice, and then you're going to be using the two different.
You end up with two populations.
So you have two separate populations, and then the thing you're going to sell, you mix those generations and you sell that seed. So they're highly inbred, but then you mix the two inbred.
Yeah. So your two different populations are you can have two or more, but they're usually known as heterotic pools. So, and you really do, and you should, if you're doing your job right, you make a bit of an effort to avoid them ever mixing in the breeding program. So they can be different things, they can be different kinds of populations, and from those two heterotic pools, you develop your inbred lines. So you do a lot of work and you make them homozygous. And this is another place where the speed breeding protocol can be really useful, because if you take an individual, it's usually especially of an outbreeding species. Again, I'm going to use maize as an example because this is really advanced in maize. So if anybody's interested, they can look up maize breeding and they'll find a lot of literature on this. And then you start to create an inbred maize. And initially, when they started doing this a century ago, the inbreds were so weak that they needed to do a thing called a triple cross, because the mothers couldn't produce enough seeds for commercial production. So they'd cross two plants to create a slightly less inbred individual, and then they'd pollinate that with a male. That's getting a bit complicated, but basically you make an effort to create an inbred from heterotic pool A and an inbred from heterotic pool B, and you usually decide that one pool is going to be your mother pool, and then you select for traits which are important also in the female line, and then the other pool is going to be your father pool, and you select for traits that are maybe more important in the male line, and some are important in both, but for example in the male line you'd want to make sure that there's good pollen production, and in the female line good ovule production for seeds, right? And then you put them together in a field and you get them to cross. Now, in the case of maize, that's actually quite convenient because the male and the female flowers are on separate parts of the plant. So if you cut off the top of the female plants, you remove the male flowers, and then they can only be hybrids because there's only pollen coming from the male plant, which still has its male flowers, while the mothers don't. And then you make your F1 hybrids. And now you've got this really nice situation where you have very uniform seeds, which the farmer needs for mechanized agriculture. He wants the plants to flower at the same time so that he can come in and harvest them at the same time. But they're highly heterozygous, so they have that heterotic hybrid vigor. And then there's another benefit, which isn't always talked about so much, which is that if the farmer saves the seeds of the F1, they will segregate, meaning that the two parental genomes will recombine and the grandchildren will all look different from one another. So that uniformity that is present in the F1 is completely lost in the F2. So if the farmer saves the seeds, and they're free to do that if they choose to do so, the plants will flower at different times. It'll make mechanized harvesting of the crop very complicated. And so the farmers don't want that, and the F1 seeds provide them with such value that they're willing to go back to the breeder every year and buy another batch of seeds. Whereby if it's an inbred like wheat, the farmers can keep the seeds and they'll get the same crop the next year. And the same goes for rice in some cases. So there's a lot of interest in the breeding community to come up with ways to develop F1 hybrids, even though to some extent, like you already mentioned, it does have this added complication that you need two breeding pools, two populations, but it provides you with this intrinsic intellectual property protection, which means that even if you're not able to successfully enforce plant variety rights, the plant kind of does it for you.
Right. And so one question I have here is, you're looking at a whole bunch of different traits. So you're looking, as we said, at the size, the protein content, the flavor, a few different characteristics. How do you know that these characteristics aren't going to be antagonistic? So, for instance, you could have a situation where one of the properties you simply don't get along with the other, let's say. So how do you know that's not going to be the case?
To some extent, you don't, at least when you're starting out, if you know nothing about the crop. What you're talking about are basically trait correlations and maybe negative correlations, or negative pleiotropy would be one of the terms. So you could imagine a situation, let's say in the case of Aardaker, where when you increase yield, protein content goes down because the plant is investing more in just making tubers with a lower dry matter content and more water in them. A good example actually of that is tomatoes. So in Europe, in the Netherlands, in the 1990s, 80s and 90s, the Dutch tomatoes started to get a bad reputation in Germany. They were called Wasserbaum because they were just tasteless bags of water. And that's because if you breed it for yield, and your yield is fruit yield, and just only focus on yield and nothing else, the easiest way for the plant to solve that problem is to just put more water in the fruit. So you end up with really high yielding tomatoes that are insipid. So this became a problem, and then the breeders got complaints from the growers. Well, the growers got complaints from consumers, and everybody realized this was a problem. And now we have, thanks to active breeding work, if you go into a Dutch or a German supermarket now, you can buy really tasty tomatoes. So they made an effort to improve flavor and incorporate that into their breeding program. It did come to some extent at the expense of yield, if you just take fruit as yield, because water is very easy for the plant to pump into the fruit. So you can have those negative pleiotropy or negative correlations, and sometimes they're intrinsic like that, right? It's just, if you want more sugar in there instead of water, the yield potential isn't the same as just pumping a bag full of water, right? You can do that very easily. But there are other cases where it might be that you can break that correlation, because it may not be in a physiological correlation like the one I just talked about, like water versus yield and versus flavor. It might be that they're closely linked on two chromosomes, and you have two alleles which are like on very close together. This one's improving yield, but this one's decreasing protein content, and then you need a recombination between the two.
So what's the recombination?
A recombination is when you get a break point on a chromosome and they shuffle. So if you were to have children, right, your mother's chromosome and your father's chromosome would line up against one another, and then they would be pulled apart in meiosis. And when they line up against one another, they cross over, and a piece, let's say, of your mother's chromosome would get combined with a piece of your father's chromosome. So that's why you get this shuffling of genetic information every generation, which is why everybody's looking different all the time. So you're getting this mixing of genetic information all the time.
And only one of the chromosomes gives rise to the characteristics you eventually see in the living organism.
Well, no, that's a different thing. That's dominance versus recessive. It's more that if you were to pass on your genetic information to your children, in most cases, the chromosomes you pass on will be part from your mother and part from your father. So they'll be 50-50. So they'll be recombined, and then your partner will also do the same from her mother and father. So your children will on average be 25% of each of their grandparents. And there's variance around that. That's the average. Whether one chromosome is expressing the phenotype versus another depends on whether you have a dominant or a recessive trait. Classically, an example of that in humans is blue versus brown eyes. If somebody has brown eyes and the other person has blue eyes, chances are their children will have brown eyes, because brown is dominant over blue in most cases. So that's if I understood your point there, that's a separate process than recombination. So you can get negative genetic linkage, and then you need to recombine them. If you do have really unfortunate trait correlations, which are really problematic, like imagine yield really does reduce protein content, there might be a situation where you can't fix that. I really hope that's not the case. I have no evidence that would be the case at the moment. And I do have some protein content data and yield data, and there doesn't seem to be sort of an unfortunate correlation between the two. There might be a moment where you have to admit defeat, to be honest.
Yeah, what would kill the program? Let's say, and where would the fail points happen in the story as well?
So there are of course many reasons why this could fail. There's not developing enough of a market, people not wanting to buy it, the protein not being good quality for a food ingredient. So those are, let's say, downstream of the breeding program. If your question is more on the breeding program itself and how could that fail, you might not have enough genetic variation. So some of our, like triticale is a relatively new crop, and it's made from combining wheat and rye. It's a really tough, really impressive crop, but for some reason it never took off. And it's because it's a synthetic crop, so it's man-made. So we combine those two genomes. And there's some suggestion that they just ran out of genetic variation and they have to go and make more of these synthetic genomes to put more genetic variation into the breeding program. And that's a lot of work. So that has maybe limited how much triticale has its potential as a crop. There are probably other reasons in this particular case to do with consumer acceptance and things like that. So in theory, that could happen, although I have a lot of genetic variation in the material I have, or at least it seems I do. So that is one thing that I'm not too concerned about. And then it could be that maybe the plant turns out, despite all of what I've said, that I just can't improve the yield. Now, I don't think that will be the case because I already see a huge amount of variation in yield in the plants that I have. And some of them, as I've said, the best genotypes would already give me 26 tons per hectare if I was to have them across the field, at least based on their individual plant level data. But I don't expect the yields this year to be anywhere near that because I'm growing such a diverse set of germplasm, and also this year the weather hasn't been great. So I'm fairly confident that will happen, but that could be one case like that. You just can't get the yields up. And there are many crops that have never fully commercialized, even though people have tried to do them, because the yields weren't good enough, and that meant that the economics was not favorable. So that can be a problem. That said, some of the most important crops that we have today are very recent inventions, for want of a better word. So blueberries, for example, have only been domesticated within the last 100 years.
Really?
Yeah, so they were only wild before. And that's a multi-billion-dollar industry. So all, like, nowadays, if you buy blueberries, they're this lovely crisp fruit. The wild ones are usually very soft. And that was one of the most important traits, like increased size, but also crisp, because it meant that they could be shipped. So now you can eat blueberries from Peru here in the Netherlands, which would not have been possible with the original wild material, because it would have come and arrived here as some sort of soup of fermented blueberries. Another example, which is, I think, also really impressive, is soy. So soy is an ancient crop, but it was not at all cultivated in the US. And it was basically a couple of people that really saw the potential of the crop in terms of improving soil quality, improving crop rotations, and providing both oil and protein. And so from the 1920s onwards, they focused on that. And within 20 years, it went from a crop which was basically not grown in the US to being one of these major crops of today. But there are, of course, many stories of crops that have failed. I'm trying to think of one now. Of course, the ones that have failed, we tend to forget. But there were a few, I think, wasn't it... I forget the name. There was a few... like crambe, which was a potential new oil crop, hasn't really worked out. Why that is the case, I'm not exactly sure. I think part of it was that other crops got sufficiently improved, like, for example, the rapeseed oil, because this would have been oil for industrial uses, not for food. So there wasn't really enough interest in trying a different crop, which is just different, but not necessarily adding additional value. And that's one of the reasons as well that I choose the Aardaker, is that none of our existing crops really have that set of traits where you have a protein-rich tuber that's nitrogen fixing. So if you choose to domesticate a new crop and it's just basically the same as something we already have, except it's a different species, that has value because we should diversify our food systems. It reduces the risks associated with disease and crop failure. But it's going to be an uphill struggle to get somebody to eat something which, from the consumer's perspective, has not necessarily any added value, but costs more.
So this was another question I wanted to ask. That is, why not, say, start with a potato and then aim for a potato with higher protein yield? Yeah.
So that is kind of what a lot of people effectively do with their crop breeding strategies, which is instead of going for new species, they try to improve what we have. And it's a very valuable approach. But if you were to use that potato example, you could breed for improved protein content in potato. But then the question would be, well, where do you get the protein from? So to get more protein, you need nitrogen. So where are you getting the nitrogen from? Yeah. So then you have to apply nitrogen to the fields. That can lead to all the problems we spoke about already to do with pollution. So then you might say, well, okay, let me make a nitrogen-fixing, protein-rich potato. And you might be able to do that with modern biotech. So there are people looking to engineer nitrogen-fixing cassava, for example, and other nitrogen-fixing wheat. So there is interest in doing that. But my point would be, well, hold on, we already have a species that does all those things. I think it's going to be easier to improve the yields of this species than it will be to engineer these new complex traits which require the development of a new organ, which in this case is the root nodule, which houses the rhizobia bacteria. The plant needs to have a very tight communication between its cells and the rhizobial cells, the bacterial cells. It needs to allocate resources to them. You're rewiring the entire gene regulatory network of the organism to provide you with a set of new traits. You're telling the potato, don't store your energy as starch, store it as protein instead. You're asking to change an awful lot of things, and I think it's going to be much easier instead to just say to the Aardaker, for example, grow bigger, because we know that works, because we've done it before. So they're not mutually exclusive. I think we should explore all avenues. I think diversity is key at all levels in our food systems, both in terms of the crops, but also in terms of the approaches we take to improve our crops and to grow our crops. But I would argue that the possibilities surrounding creating new crops from existing wild species are underexplored. And I think in many ways, especially with the advances in modern technology that I've already spoke about, it's a better bet.
So before our conversation, you mentioned that one of the things that makes the breeding program difficult is that the plant is polyploid.
Oh no, this is not polyploid.
This one's not.
No, this is diploid. Potato is polyploid.
I was going to ask, can you explain what that is?
Yeah, sure. So we are diploid, meaning we have two sets of every chromosome, one from your mother, one from your father. Potato is tetraploid, for example, which means it has 4 sets of chromosomes, 2 from the mother, 2 from the father. In the case of potato, it's what's called an autotetraploid. So it basically, the original potato genome doubled from a diploid to a tetraploid. You can also have another...
How does that happen? Sorry.
It happens by accident.
Just how...
So you might get an incomplete meiosis. So it happens, like if you just take a piece of pollen, like a sample of pollen from a plant, and you look at all the pollen, sometimes you'll see some pollen grains that are twice as big as the others. And that would be basically incomplete meiosis. So there wasn't a complete reduction in the chromosome number, and you now have diploid pollen. Now, if that diploid pollen happens to land on a stigma, the female part of the plant, that also has an egg that also didn't do its meiosis properly and has a double, and you get 2 doubled sets, and you get a tetraploid. You can also have it that the tetraploidization happens in mitosis, so you get a doubling of the meristematic tissue, and then you just get a tetraploid plant arising. And that's how we make them in the lab often, which is you take an extract from, and this is kind of funny, it's like hocus pocus, but you take autumn crocus, right? So this is a species of plant, autumn crocus, and it has a compound in it called colchicine, which is very toxic, by the way. And if you apply that to the meristem of a plant, it messes up its meiosis and its chromosomes will double. So you can make a polyploid out of a diploid by just putting drops of this extract of autumn crocus. And people did that a lot.
Why would you want this?
Because polyploids are usually bigger than diploids. They have twice their genome size, twice their number of chromosomes. The cells are bigger, and they are often bigger. So you do get a lot of...
the plant itself, you get bigger yield.
Often you get bigger yields, you get more vigorous plants, and it also allows you to combine genomes of different species.
Could we do this with a human?
So, good question. Probably not. For some reason, plants are very tolerant to polyploidization, but animals are not. So, certainly mammals. Polyploids in mammals are very rare. I think there's a couple of rodent species that might be polyploid. I'm not an expert on animal genetics.
We can get extra chromosomes though, right? So
that's called aneuploidy. So there's a difference between polyploidy and aneuploidy. So polyploidy is usually extra, like a duplication, or you have 2 copies of your genome, or you have 3 or 6 or whatever, but you have the whole genome replicated multiple times, right? And you have species like sugarcane, which have, I don't even know how many copies of the genome sugarcane has, and it's even variable. It's crazy mess, complicated genome. But then you have aneuploids, and that's when only 1 of the chromosomes or 2 of the chromosomes is duplicated or missing. And a classic example in human is Down syndrome, where you have an extra copy of chromosome, I think it's 21 or 22. I'm not sure. Don't quote me on that. But then you end up with it's not great for you. And it's the least bad aneuploid in humans because you're still viable. Most aneuploids, they die before birth in the case of humans. And aneuploidy is often a problem in plants too, because you get an uneven dosage of your chromosomes. So you have one chromosome that's overrepresented or underrepresented, and then you get issues. Whole genome duplication is a different thing, and for some reason plants are quite tolerant to that.
Because the ratio is correct.
Yeah, well, that's what we think, yes. And it's certainly why they're more tolerant to that than aneuploidy. Why they're more tolerant to whole genome duplication than we are, I don't know that we know why that is the case. Maybe somebody does, but I'm not aware of if we know why mammals don't tolerate that, but plants do. That said, in our deep evolutionary history, there were some whole genome duplication events. So in a way, we are ancient. At an ancient level, we are polyploids. But after polyploidization, there's a gradual process of rediploidization. This is in the evolutionary span of things. So over time...
So mammals used to be polyploid at some point.
One of our ancestors long ago, I don't know, hundreds of millions of years ago, was a polyploid. And it probably... So polyploidization events can lead to genetic innovation as well. So one of the examples that I'm aware of is in the Brassicaceae, that's the cabbage family. There was a polyploidization event which then allowed for a diversification of the secondary metabolites, which gave rise to more diverse glucosinolates. So glucosinolates are these compounds that give you that mustardy smell. So when you eat mustard and you get that, or wasabi, and you get that burn in your nose, those are glucosinolates. And the plant has them because they prevent animals from eating them. Or in our case, encourage us to eat them. But, humans are strange. So, but
then we breed them, so it's very successful for them.
Yeah, in a way, yes. Good point. Very, very valid point. So the genome duplications in brassicas have actually allowed for what's called subfunctionalization. So you get extra copies of these genes, and then because you have extra copies, some of them can take on new roles, and then you can get more complex secondary metabolite profiles and evolution of new kinds of, in this case, glucosinolates. But generally what happens is after you become a polyploid, having extra copies of all your genes isn't necessarily a great thing. So over time, those extra copies are lost.
Because energy, you need more energy.
Yeah, or just regulating the whole thing, or even just combining. Like, for example, potato has a lot of sterility issues because combining and doing that meiosis we spoke about earlier. So I just talked about...
The recombination.
Yeah, well, I talked about your mother and your father's chromosome lining up and then pulling apart. But, like 3's a crowd, 4's even more of a crowd. So when all 4 chromosomes are trying to line up against one another, you get a thing called a tetravalent. And sometimes then meiosis just doesn't work. And then what happens is you get uneven numbers of chromosomes in the different gametes, and you get aneuploids. And aneuploids, as we just spoke about, are pretty sick. So you can get, especially with autopolyploids, that's when the potato genome duplicated, they have a lot of problems and can have a lot of problems with sterility, which isn't great. But over time, they evolve away from that. And one of the ways of evolving improved fertility is gradually going back to a more diploid-like state. So, for example, soybean was long ago a polyploid, and now it's what we would call a mesoploid. So it's on the way back to becoming a diploid, which is a gradual process that takes millions of years.
But I don't quite understand that. So to me, it sounds like you can either have 4 or 2, but...
So yeah, the rediploidization event is a gradual process where you're no longer like chucking out chromosomes, but you're chucking out genes and pieces, and then gradually maybe even sometimes you get the chromosomes refused together and you get a new chromosome
that's just twice as long or something.
Yeah, or 1.5 times as long because before it fused it already lost a bit, and then gradually you go back to a new karyotype being the number of chromosomes in a species. So our karyotype is 23, then you would have something which is now basically behaving like a diploid, but only when you do special kinds of analysis you can see, there must have been X numbers of generations ago a polyploidization event, which is why we can see in our own genome the legacy of an ancient polyploidy event, genome duplication event.
So I don't quite understand this. So how do we know, how do we pull out that history? How do you know that there was an event, say 10,000 years ago or
much longer,
or many generations ago?
Yeah.
Like how do you spot that in a genome?
Yeah, so I'll be honest and say I'm not the right person to answer this, because this is like special kinds of like bioinformatics that people do. But from my understanding, what they do is they can look. So mutations tend to occur at a relatively fixed rate, which is known as a molecular clock, which is kind of how we can date these things. And they can look across the genome and they can sort of see, well, it looks like there's an enrichment of duplicated genes, which based on their level of divergence means that it must have happened X numbers of generations ago. And then we can infer from this that there must have been a whole genome duplication at this point. And then you can look at multiple species, right? Because we share a common ancestor with all the other mammals. And you can sort of say, well, they're all showing this signal that long ago there was a whole genome duplication event. Now, most of those excess genes have been subsequently lost, so we're down to a single copy for most of them. But the few were retained. And because you got 20,000, 30,000 genes, there's enough signal there for you to be able to still pick that up. And of course, eventually over time, that signal might eventually be lost. So maybe back when we were amoeba or something, there was another whole genome duplication event, and maybe we no longer have the ability to pick up that signal. I don't know. But yeah.
So one aspect of this that I don't fully understand is I would think that so the story could be one of two things, as I imagine it. You could have a baseline, so you could have sort of like a frozen sample of a plant, say from 10,000, 100,000, a million years, something like, I don't know how long DNA lasts, but you could have a frozen plant that then you can compare to the modern variant and see, how many changes have occurred during that time and so on. Or maybe it is that you need to look at the full population rather than individual plants. So how do you pull out I don't understand how you could do this from a single plant.
From a single plant, you could do it, the polyploidization, I think you can pick it up from a single individual.
Okay.
Because you can see that there's a cohort of genes which clearly have been duplicated, and based on their divergence...
Oh, you see the recombinant...
Right. You see that, like, let's say there's 30,000 genes, and then you see of those 30,000, there's a subset, I don't know, if you pick your number, a few thousand, which clearly are ancient duplication events of one another. And based on the number of sequence divergences between each one of those duplicates, you can say this all happened at the same time. So it must have been an ancient whole genome duplication event.
Okay, so you're saying, maybe I'm misunderstanding, but you're saying that at some point you were a polyploid, and you lost a few genes, and then they recombined into so that you now have a diploid, and you can see that the diploid has duplication along the DNA. You say there's 30,000 that are the same here and 30,000.
No, maybe I didn't explain this very well. I don't know if that's exactly how they do it. I must admit this is not my area of expertise, but it's more that. So first of all, when you do a whole genome duplication, having extra copies of some genes is actually not a great thing. And you want to get rid of those. So those are lost pretty quick. And this has been confirmed empirically. So people have made polyploids, and then they have a lot of instability and issues at a genomic level. And then they keep selecting them for several generations, looking for ones that are more fertile and more stable. And they see certain genes being lost rather quickly. And that then allows the plant to balance, because there's these complex gene regulatory networks, and if you have an imbalance, that's not a great thing. Over the longer evolutionary timescales, more and more genes are gradually lost, whether it's because they're a problem or because just having one copy is all you need, and by chance one of them gets lost. So the number of duplicated genes from an ancient polyploidization event declines over time, and that process leads to what we call rediploidization. And then you also may get some of the chromosomes fusing back together and get a smaller chromosome count, because you may go from, let's say your original was 10 and then you doubled to 20, and then over time you may go back to, 15 or even less than 10 potentially. There's no reason why not. But still, there's that legacy still there in the signal, because some genes, the extra copies were retained because maybe they were beneficial. Maybe it gave you that new metabolite. Maybe it gave, and there's enough of those that you can pick up that signal that, ancient, long ago, there's so many genes duplicated that we can still see that. And in our history, and certainly in the histories of certain plant lineages, you can look at these genomes and say, well, approximately 2 million years ago, there was a whole genome duplication event. But there was also a whole genome duplication event 100 million years ago. And that's at the base of some radiation. You can see that all of one clade of plants have that. And then you can see, and often these whole genome duplication events, they occur from a phylogenetic perspective at key points and lead to an awful lot of innovation and diversification in long-term evolutionary lineages. Now, back to the original question as to why you would do this in a plant breeding program. So some people think and argue that this is a really exciting way to do genetics, which is to make polyploids. Now, people have thought this for a long time. The Josta berry that we spoke about, that's growing around here. This is a man-made species, which is a multi-interspecies cross between, I think, North American blackberries, European blackberries, and
you mean
gooseberries.
Outside of the conversation?
Yeah. Like literally, yeah, exactly. We spoke about it before. That was made using polyploidization. So the addition of colchicine that was done in the 50s in Germany, and this was a really hot topic back then. It lost a little bit of favor over time. People didn't get so focused on polyploids, but just this year there was a big announcement from a guy known as David Friedberg. He's famous for having the only ever unicorn in the agtech space, which was the Climate Corp, bought by Monsanto, 10 years ago for $1 billion. And he's now into polyploid breeding, and he calls it boosted breeding. And his argument is that because you've got extra copies of the genome, there's lots of additional benefits associated with that. And some of that is true. It's not as new, of course, as they would like to make it out to be, but they have to, right? They want to get investors on board. And other people have done this successfully. And I think it'll work very well for crops like potatoes. I don't know how well it will work for some other crops which are not normally polyploids, because of the fact that you can get sterility issues. Now, that's not an issue with potatoes because you don't need to harvest seeds. You only need to harvest tubers. But it is an issue for certain seed crops. That said, some of our most important seed crops, like wheat, is a polyploid. Wheat is a hexaploid. It's got 6 copies of its genome. But it's different than potato because it's an allopolyploid. So an autopolyploid means the one genome was duplicated. A allopolyploid, it's actually, there's 3 genomes in wheat from 3 separate species. And each one of those is a diploid genome. So that's why you have 6 copies. So there's an A, a B, and a D genome. I don't know what happened to C, but for some reason they skipped it. And that is So it's a hybrid of 3 different species. Yeah, so what happened was A and B combined, and that made a tetraploid, and that's Durham wheat. That's what we get our pasta from. And then some point, and that happened long ago, like millions of years ago from what I understand. And we then domesticated that species. And then very early on in the domestication of wheat, maybe longer ago, I don't know if we know for sure exactly when this happened, there was an additional hybridization event with the D genome, which is with a type of goat grass, and that gave rise to bread wheat. So Triticum aestivum. So that's the most important wheat crop that there is. There's several wheat species that we grow, but the most important one is the hexaploid. And that is interesting in that this weird genetic accident has given rise to one of the most important crops in existence today. So polyploid breeding definitely has potential, and it's very interesting. It's not something that I'm looking at with regards to the Aardaker yet. I wouldn't say that I wouldn't do it, but it's certainly not a first port of call for me because there's lots of other work to be done first. But so the original question was going to be, is life easier for you because, well, now the plant is a diploid? Or yeah, so from an analysis perspective and from understanding how the genetics is going to work out, it's certainly easier to work in a diploid because there's just it's less of a mess. Potato breeding is complicated by the fact that you have these 4 different chromosomes and they recombine in weird ways, but it's also complicated because you have a lot of inbreeding depression and a lot of problems with fertility, so not all crosses are possible. So yeah, potato breeding as a whole has struggled with this. So, but if you ask David Friedberg and you go to Ohalo Genetics, the boosted breeding is going to solve all of their problems. We will see. Solinta, you have actually taken the opposite approach, and they've decided to make potato back into a diploid and breed that way. And they've been working on this for a while, and they have now products on the market, and they have a lot of success. So I mentioned earlier about the importance of potato as a seed crop. That is definitely one of the advantages that both Ohalo and Solinta go for, but they are also saying that they're simplifying the breeding process. And Solinta is by doing that by making it back into a diploid.
I see. So they want to sell the seed, and
they're also they say that we can breed better than before, which is, I think, to some extent true.
And from a sustainability perspective, this makes a lot of sense, as you said, right? In my supply chain, I don't need to cut around 1 million tubers. I can just sell the seed.
That for sure. And then another thing is that breeding itself is probably one of the most sustainable innovations you can get, right? Because, look, we need to eat food, but our food system as a whole is both a victim of all the environmental damage around the world, climate change, but it's also a perpetrator, right? 33% of global emissions are due to the food system as a whole. It's the main source of land use change, main source of water pollution from phosphorus and nitrogen. So this whole planetary boundaries framework, where we look at the planet as 9 major systems, the main driver for pushing us over the edge of the ones, I think we're over the edge of 6 of the 9 at the moment. And I think for 4 of those 6,
this
it's
is the donut economics.
No, this is the Stockholm Resilience Institute. They have this thing called the planetary boundaries framework. And it looks like a pie chart, and they divide the world up into all these, the Earth into these major systems. So you have like ozone layer, CO2 emissions, biodiversity loss, land use change, biogeochemical flows of phosphorus and nitrogen, novel entities, and there's others that I just don't remember off the top of my head. But food is really driving us over the edge on a lot of these. But if you can improve the crops genetically, that is a lasting impact on the sustainability of food production. And it is intrinsically sustainable, because it's not like you need to throw new chemicals on the field, or you need to add more fertilizer or do more intensive plowing or whatever it is that might improve your yields, but then it may have a negative consequence in terms of the health of the soil or the sustainability of the ecosystem as a whole. If you can just improve the plant, that's a lasting, because it's heritable, it's passed on to the next generation, and it just intrinsically sustainable. I think breeding is one of the most sustainable ways to improve our food supply. Which is why, of course, I have skin in the game. Maybe I'm a bit excited by it.
But okay, so in terms of raw numbers, so at the end of the day, after you finish your breeding, well, maybe it's
you're never finished.
You're never finished.
You're never finished.
But in the
great for job security.
Let's say in the next 10 years, what do you think in terms of raw numbers in comparison to a potato, in comparison to a soybean? What do you think you can actually deliver realistically? And let's say what's the ceiling that you hope for, but then the average plant that you pull out of the ground?
So the ceiling, it's hard to say, right? I don't see any reason why this cannot eventually reach yields similar to or better than potato, which would then be an average of 40 tons a hectare for people to understand, in the Netherlands at least. And the best farmers can pull out 80 tons a hectare from a potato crop. That would be something that I would hope for in the long run, and then we would have incredible yield potential from a protein perspective. How long it's going to take me to get there, I don't know yet, because I don't have enough data. I need to do this for a few years, and then I will be able to answer that question with a margin of error. But right now, this is the first year that we do field scale production, so I just don't know. What I would be hoping for that is within 10 years, we'd be at least aiming for something like 20 tons per hectare. And that would already give us that 40% more protein per hectare than soy, an average. If we can get the speed breeding running and the genomic prediction running, I'm relatively confident we'll reach those numbers. But it's always hard, because there's a lot of unknowns, and there's still some data that I just don't have. So it means that there's some projections which, as a scientist, I'm a bit uncomfortable with, but at least I think that should be reasonable. And then that would already make it, I think, a pretty profitable crop, because of the fact that you could hopefully produce If you can produce more protein per hectare, you should be able to produce it at a better price. Quite simply, because one of the biggest overheads in your inputs is just space, land, right? And all of the inputs that come with that, and the fact that you don't need to add nitrogen fertilizer. So that would be the hope.
So for the investor that doesn't care at all about the environment and they've no interest in sort of the bigger mission, how do you sell this? So the economics...
Oh, I don't sell to that kind of investor. No, but I'm being facetious here, but that is a serious point. You do need to be aligned with your investors. Like, it's a separate point, but okay, so for a thought experiment, let's just imagine that they don't care, and then, go on.
So just economically.
Yeah.
So what is the sell here? Like, what's the earning potential of this crop?
Yeah, so here in this part of the world, in Europe, farmers are very picky about the crops they grow because the land is really expensive, right? So your crop really needs to be profitable. And there are some crops like chicory, for example, where there's demand, but it's not profitable enough for the farmer, so the farmers stop growing them. So we actually have problems with chicory inulin supply because they don't make enough money. Now, market should solve that, you would say, but maybe not. So there are different ways of improving the profitability. One would be to move away from a place like Northwest Europe where land is expensive and go somewhere where land is cheaper. And certainly that in the long run, it'd be something I'd like to do with the Aardaker. And it should grow very well in, for example, the plains of Canada and America, where input costs are much lower. Certainly land is much cheaper. And because it grows very well in Russia and
it ships well?
Well, if you're going for food ingredients, you can have on-site production of the thing, and then you would be shipping just the dried protein powder or starch. So in that case, that shouldn't be an issue. For fresh consumption, you'd probably want to do it closer to the consumer because just like with potatoes, otherwise you're shipping a lot of water, right? Which is not great. But it does store well. I mean, I've had tubers that I've stored for up to 20 months. They're still viable. As long as you don't let them dry out and keep them cool, they're fine.
So it is similar to a potato in that regard.
Yeah, perhaps even better in some sense, in that they...
I guess these are much smaller though.
As I said, I've had some that are up to 500 grams, so you can have big tubers.
I see.
These particular ones are small. I didn't happen to have one of those big ones. If I did, I would have brought it along.
But these are choice, right? This is
we'll say that. So your question about what is the financial pitch? Because there's a few things. One, if we can get up to like 20 tons a hectare and then produce 40% more protein than soy, even as a food ingredient, this should be profitable. There's the other advantages, which that, let's say the farmer is a potato farmer, right? And they've already got the machinery and they make their money on potatoes and maybe onions in this part of the world. And then they have to grow what they actually call it a rest crop, something like wheat or barley. And they make hardly any money on those, but they have to grow them, otherwise they'll get too much disease. So if you could go to them and say, I have another crop for you, which is going to be profitable, like your potatoes and your onions in your rotation, but it's even better, it's going to improve your soil, it's going to give nitrogen to your soil, so it's going to improve the quality of the soil for your follow-on crop, whether that's potato or whatever. It has great yields, and there's a lot of demand because we need plant-based proteins.
And you can use the same equipment.
You can use the same equipment. You don't need any new investment. But one extra benefit over, let's say, your potato is the above-ground biomass. You can just cut that and sell that as a high-quality animal feed. That in and of itself may not be a lot of money, but farming is a low-margin business. So if you get an extra €100 or €200 off your hectare, that might pay for all of your planting activities. So now you just improve your margins, and that's kind of what I'm going for here. So at the moment, it's profitable because it's a premium product, right? So we're able to charge a good price for the tubers themselves, which means that we're able, because it's also, but it's a very limited production, right? But we want to go to scale because scale is where impact happens. And then you need to move from premium product to commodity. Most businesses would never want to do that because your profit margin goes down, but your scale goes up, and that's ultimately in the world of agriculture. That's, again, as I've already said, that's where the impact comes from. And that's what I really care about, right? Like back when I was in COVID times thinking, how can I help? If I'm being completely honest, if we stay in the realm of Michelin star restaurants, we're not really changing anything. It's nice, it's cool,
you want to sell millions of these.
Yeah, large volumes, large acreages, because if this is on the acre and we're able to produce more protein per acre, that means we need less acres for the protein. And then this comes to this land sharing versus land sparing
You can
debate.
rewild.
You can leave more space for nature, and that's something that's deeply important to me. I would wish that more of the planet was self-willed land, so wild. And I think that's something we need to work hard on, is making sure that we can share this planet with the rest of life that's out there. And the only way we can do that is if we can produce our food on a smaller physical and environmental footprint. And there are many ways to do that, one of which is indoor farming, and that's a space I've explored that has challenges associated with it. But one is to try and create crops which have lower input but higher output, which is a hard thing to achieve, but I think this crop can do that because you don't need to add the nitrogen, and because it's a tuber, it has better yield potential than our other existing protein crops. So hopefully we can one day have a crop which is just that, lower inputs, higher outputs, which improves the sustainability of food production, in this particular case, all over.
One of the nice things I like about this program is that if you're successful, let's say everything goes ahead as you would like, it might be the case that in 1,000 years, 2,000 years, people are eating this crop that you developed. Right?
Yeah.
That's the potato on the plate.
Yeah.
So long after
I'm gone, dead, forgotten. Yeah, that. I have to admit, that's a fun aspect of it. One aspect that's
the being
also
dead and gone.
Oh yeah. Well, the fact that there's a potentially persistent legacy. It doesn't matter that nobody remembers me, but I would know that could be the case. But that speaks to something which I think is nice about this work, which is that this is a useful innovation regardless of what way things go in the future. And what I mean by that is that should we be successful and we one day create a sort of, if you're a techno-optimist, let's say, and you think, well, we're going to solve all our problems, we're going to have a nice equitable society in the future, civilization will continue to advance, and we're going to go to the stars, and we're going to be super, super successful and super advanced. Having a more sustainable protein source and being able to produce it in fields is still useful, even in that scenario of really advanced society. Maybe one day we won't need it. Maybe we'll all just be machines or whatever. But within reason, it's still a useful solution for at least the foreseeable future. But should we go the other way, which is that unfortunately we don't organize ourselves, and who knows what, there's an ecological disaster, there's wars, there's so many things that could go wrong, civilization collapses, this is also still useful because you don't need high tech to be able to grow that. But if the breeding's been done with all modern technology and we have something which is resilient and high yielding and protein rich and nutritious, it's basically a protein rich potato. So even in a pessimistic future where people go back to sort of, let's say, the Middle Ages or something like that, this is still useful.
We're back in the Middle Ages, but with this.
Yeah, we have the Aardaker. It's a
persistent technology that we don't, we might get into a stage where we can't build computers anymore.
It's a durable innovation. Yes, it's a durable innovation. And it's an innovation and a contribution which is useful under a wide range of future scenarios, which for me is nice because when I worked in the indoor farming, there's always a little bit of you that thinks, well, if the shit really hits the fan and we all go down, then all this work is kind of useless because we won't have the tech to be able to continue doing indoor farming. But that's not the case with this. So there's a little bit of me that, of course, there's a risk that I fail completely and this crop just doesn't work out. But if it does work out, it's just useful under such a wide range of future scenarios that I feel like my time is well spent, which may be a bit self-indulgent, but still, it's nice to think that you're doing something which is going to be useful to some extent, no matter what.
So what makes you happy? Because so to give some context to the question, at the beginning of the conversation, you said that you were worried about the environment, you're worried about the impact we're having on the planet, potentially climate change fears, this sort of thing. What makes you happy and what makes you optimistic?
Carrots. Carrots make me happy and optimistic when I'm thinking about the Aardaker. And that's because, have you ever seen a wild carrot?
No.
Well, there's some growing around out here. After this, we're going to go outside and find a wild carrot and I'll dig it up for you. You would have no idea that was a carrot based on the root, except for the way that it smells. So the moment you break the root, you'll smell carrot. But it's just normal looking root with sort of white, spindly, forked, woody, completely unattractive. But here in the Netherlands, they have these things called winterpelen, these gigantic carrots. You can buy them in the supermarket. They can be sort of as long as your forearm and several centimeters thick. And they've all been developed mostly within the past few hundred years.
And they're tasty at that
They're
size.
tasty. There's this incredible yield difference compared to the wild thing. And whenever I look at that, whenever I'm peeling a carrot, I always think, if they could do that with carrots, and considering the starting point of Aardaker is already a much better starting point, this is much better than a wild carrot, it makes me very optimistic that I can succeed with Aardaker. So I don't think that's really answering the question. I think you were looking for something broader about society, but definitely when I'm peeling a carrot, I'm always like, yes, okay, you're not crazy. You might actually make this work.
The question was more designed to get a feeling for who you are as a person.
I don't know. What does that say, that he likes carrots? For me, what makes me optimistic as a whole, well, there's a lot to be pessimistic about, let's be honest, right? And the things that worry me are more about politics and I do think COVID is a great example. We had the technology to deal with COVID, and in the end, we developed a whole new type of vaccine in record time. From a scientific standpoint, it was incredible what we achieved, but most of the suffering and mess-ups came because of the political systems we have. There's a great quote, which I'm going to destroy, but I forget, was it who said it? Maybe you know the one where it's like, I think it was Carl Sagan maybe said it. The problem is that we have godlike technology, but Stone Age brains and medieval political systems. You will have to look.
I know the quote. I can put it on screen.
Yeah, you know the quote. I forget it, but I think that is very true, and that is both a source of optimism because of the technology and what we can do, and what I, for example, hope to do with Aardaker. And I can do that. We haven't spoken about biotech or genome editing yet, but I can do that without going down that route already, just because of the incredible computational powers we have, and because we can interrogate and understand genomes in ways that were never possible before. But on the other hand, classic, like the truism, with great power comes great responsibility. And I don't think as a global civilization we are fully aware or owning that responsibility. And there are wars today between people. That's just madness. Countries are invading one another, and all these things are happening, and all this, the older I get, the more I realize geopolitics and stuff, it's really just like kids in a schoolyard just looking for dominance over one another. It's just mad that that's how things go at a large scale. So that worries me. So what makes me optimistic? That there are people out there trying to make the world a better place, and that they are now having the tools to do that. And I suppose the problem, and both the I suppose what I'm getting at here is that the optimism and the pessimism are two sides of the same coin. The thing that will save us is also the thing that could destroy us, which is our ability to develop technology. So AI, really topical at the moment, that can solve so many problems, but it could also be an absolute, and I think you've had podcast episodes on this, it could also lead to real disasters. Are we going to manage these things? And I think the thing that scares me is that our current political systems are just not equipped for it. And I'm not saying I have a solution. I don't.
But you have a tool to
I
make
have
it easier.
something that I think can make at least our food production more reliable. And I hope that more people out there will just think, what can I do? What am I good at? Whatever that is. I mean, I choose genetics and breeding, but it could be, for example, science communication, like what you're doing here, which would hopefully educate more people and reduce ignorance and misinformation. And from that comes an awful lot of, misinformation and ignorance give rise to a lot of bad things in society. And there are so many things that people can do and work towards. So I think if you reflect and ask yourself, what am I good at? What am I interested in? And how can I leverage those interests and passions towards something which might create a better outcome for the future? Yeah, bit by bit, we can create a better world. And there are a lot of people doing that, right? And there's so much diversity in what people are doing, and there's so many ways that people can do that. obviously not just crops. So yeah, that makes me optimistic as well. That and carrots.
Well, Padraic Flood, it's been a pleasure.
Thank you very much. Thanks a lot for having me. It was really fun.
Cheers.
So we're just out looking for a wild carrot. And we have one here, conveniently growing on the side of the road. And you know it's a wild carrot because it's got this interesting thing. It's got this umbel. It's classic for carrots. And it always has one purple red flower in the middle. I don't know exactly why. I think it's to invite more insects there because it looks like there's already an insect on the flower. This is a normal carrot. We didn't manage to find one of the big ones from the super. Well, we didn't get around bothering to buy one, but this is it. So let's just dig this up and have a look. All right, well, this is actually much better than they usually are, to be honest.
It's almost as big as the...
exactly. But normally they're really crappy. So this is actually one of the best wild carrots I've ever dug up.
breeding this.
Yeah, this is... I'm sorry that I've ended this genetic lineage now, but just smell it.
Exactly like
It's
carrot.
just like a carrot, yeah, but it's white. So if we break that, like, you can see these are the things we've changed. So the flesh is completely white. I really want to dig up another one now just to prove that they're normally really, really small. But this is a really good wild carrot, and this is a pretty poor domestic one. I feel like we've undersold here massively. But anyway, generally, this is very woody as well, like really woody. It's actually the whole center here is just wood. You see that? That's like really woody. But this thing, well, everybody knows what a normal carrot is like. So... yeah, I find that very inspiring.
Is it tasty, the wild? Because it smells great. It smells nice.
Well, originally it's thought it was domesticated as this flavoring of stews. Yeah. So, but it's, I mean, this one's not very clean, so I don't really feel like munching on it right now. But in general, they're not as nice as the domestic ones. I'll try and just, it's really tough. I mean, it tastes like a bad carrot, you know, like no sweetness, but carroty.
All right, so hold them up together. We'll see. So an excellent wild carrot and a really terrible
domesticated
one.
Yeah, you can see this was clearly not rehearsed.